The overall goal of the following protocol is to measure the metabolic profile of zebrafish embryos in response to specific genetic or physiologic alterations. This is achieved by first modulating a particular genetic or physiological pathway using a selective pharmacologic compound. Next, when the desired developmental stage is reached, basal respiration is measured in control and treated embryos using the Seahorse XF24 analyzer. Then respiration parameters are calculated from the data. Results are obtained that reveal that metabolic profiles can be studied in the developing zebrafish in vivo, and that these profiles can be modulated by targeting a specific gene during embryonic development.
The main advantage of these techniques, in regards with already available techniques like cell-based assay or indirect measurement of respiration, is that we are dealing with the whole organism, a whole embryo, allowing us to look at all the aspects of mitochondrial respiration.
This technique can help answer key questions in metabolism by measuring whole animal respiration and mitochondrial function.
To begin treatment, collect zebrafish embryos after fertilization. For in situ hybridization or oil red O staining, add 0.2 millimolar 1-phenyl-2-thiourea to prevent pigment formation. Incubate the embryos for about 26 hours. Add the desired chemical at an appropriate final concentration with a vehicle-only control. Before using the Seahorse XF24 analyzer, run the calibration that houses the oxygen and hydrogen fluorophores. To prepare the 24-well XF24